Good morning, good afternoon, good evening to our online visitors and participants. I believe we have about 140 online now, and to the people present here at Afrigen. This is a great event for us. It is the science of mRNA that will take us forward, and for this modality and this technology to meet its true public health benefit. And today is about the science. It's about having some of the best in the world in the room, sharing knowledge and building the knowledge base of the African continent and the low and middle income countries that's so much part of this program. This is a delightful day for us, and I welcome you all and looking forward to the proceedings of this third Science Colloquium of the mRNA Technology Transfer Program. Thank you. Over to Charles.
Thank you very much indeed, Petro, and I would like to add my welcome to yours. I am standing in for Martin Frieda, who is on his way, and will be here by tomorrow morning. So apologies on his behalf that he's not here to do this. So we have a really great program today with a number of presentations to start with, and then we got a panel. And I hope that that will give some time for anyone online or here in the room to ask questions. But it is pretty packed, so we'll see how we go. So we're going to start off with a video message from Drew Weissman, who, as you know, won the joint Nobel Prize just a month ago, and who's been working on mRNA for some good 20 years. So over to Drew.
Thank you very much. I apologize that I can't be there in person.
Thank you very much. I apologize that I can't be there in person.
I'm very happy.
Life has been complicated lately. I'm very happy to talk to and introduce this seminar series. I think what's critical to understand is that Katie and I started this work 27 years ago. And from the beginning, my goal has always been to include the entire world, to allow equity across the world, to give access to science, to mRNA, to vaccines, to therapeutics. And with Afrigens and the WHO and many other groups' help, we've been able to accomplish this. So the mRNA revolution, which actually started quite a years before the COVID-19 vaccines came out, has mostly centered on vaccines. And the wild success of the COVID-19 vaccines, about 95% efficacy in phase 3 trial, show the power of this vaccine platform. It's currently being used for many different diseases, many different infectious diseases, from viruses like HIV, hepatitis C, norovirus, Zika, other flaviviruses, many different viruses. But there's also applications in cancer. We've seen a couple of results from clinical trials showing success in melanoma and pancreatic cancer. And new developments involve making vaccines for autoimmune diseases and food and environmental allergies. So the wide-ranging applications of the RNA technology are really enormous. And this is something that the whole world needs to do. My thinking of this is that regions have diseases that are specific for their areas. In order to allow local regions to develop therapeutics they need, that they have to have access to the RNA scientific technology and to the ability to produce GMP vaccines, which is what's been accomplished through this WHO program. The future of RNA is also quite enormous. It's currently being used as a therapeutic protein delivery system. So we and others have been delivering RNA to the brain, to the heart, lungs, T cells, bone marrow stem cells, and many others. And the big difference here is that current gene therapy approaches involve taking cells out of a person, putting them in culture, infecting them with a virus that encodes gene editing technology that fixes the gene defect, and then putting those cells back in. Such a therapy is about to be approved by the FDA in the United States for sickle cell. The problem is that this requires extensive clinical and research facilities that are not available across the world. And it's likely going to cost in the region of $1 to $4 million a person. When you've got 300,000 people a year being born with sickle cell anemia, the numbers don't add up. So the approach that we've been taking is in vivo gene therapy. And what that means is that we use RNA LNPs and we target the LNPs to the cells of interest. We've been able to deliver these LNPs to bone marrow stem cells and deliver gene editing technology. So we can fix the defect in sickle cell anemia in the body with a simple IV injection of off-the-shelf RNA LNPs. That can be easily given across the world by a simple intravenous injection. This has wide-ranging applications to thousands of other genetic diseases of the bone marrow, as well as diseases of the liver, lung, brain, heart, and many other organs. Therapeutic protein delivery was actually one of the earliest developed technologies. Currently, there are phase 2 going into phase 3 trial using RNA to treat heart attacks and heart disease. We see this as a rapidly expanding area of research. Heart disease is the #1 killer in the world, so it's incredibly important for the entire world. By having GMP sites across the world that can produce these therapeutics and develop their own therapeutics, it opens up the possibilities for new RNA therapies. So to me, this is an incredibly exciting moment in the field of RNA research. And the development through WHO, who funded and helped Afrigen build their GMP site, I think is going to be critical to allowing wide-scale RNA use in the future. Thank you very much.
Apologies for this interesting, it's as if we've got a sort of enormous bee trapped behind the screen. Yes, very hopeful and inspirational talk from Drew there. So I'd now like to turn over to Professor Dame Sarah Gilbert, who's the Professor of Vaccinology and Pandemic Science at the University of Oxford. Sarah.
Thank you. Can you hear me through this microphone? Yeah, that seems to be working. Thank you. So my talk is entitled Assessing Impact of Access to mRNA Vaccines in Low and Middle Income Countries. And this was Petro's idea. She gave me this title, and I'm going to do my best to address it. But when I first saw the title, I thought, well, I'm not sure I'm the right person to talk about this because, I don't work on RNA vaccines. I collaborate with people who do, but that's not really my field. I don't actually work myself in low and middle income countries. And this is a really big topic. But when I thought about it, I realized that actually it's such a big topic that there's probably nobody who is the expert who should be speaking to this. And what is needed if we're going to really have impact using RNA vaccines in low and middle income countries, we're going to need a huge collaboration. We're going to need input from lots of different people. We're going to need lots of different viewpoints, lots of discussion, lots of free discussion where we can put our different ideas and try to come to solutions together. And so I do have experience that is relevant in this area. And so what I'm going to try to do briefly this afternoon is just talk you through a little bit about how I came to work on vaccine development, particularly for low middle income countries. So I've just lost my slides. Thank you. and my thoughts, which are my personal thoughts, on some of the opportunities and some of the challenges. So I'm not going to give you the answer to all of this. I'm just going to give you some things to think about, and maybe then we can talk about some of these in the panel discussion. So let's start with where I first got into outbreak pathogen vaccine development. Now, prior to 2014, when this Ebola outbreak happened in West Africa, I had been working originally on malaria vaccine development. Then I moved into flu vaccine development. And that's what I was working on in 2014, and originally came into this trying to find technologies that we could use in people to induce T-cell responses, to get protection through T-cell responses, because with very few exceptions, the vaccines that we have today that we all use, get vaccinated with, get our children vaccinated with, they're licensed because they produce antibody responses, but for some diseases, in some situations, we need to think more about the T-cell responses. So that's where I started from, but then got into testing different technologies, moved into testing viral vectored vaccines, which are great for inducing T-cell responses, but they also give you antibody responses. They actually give you a very complete immune response, which is great. And so that's what I was doing, working on pandemic potential flu prior to 2014, thinking about broadly cross-reactive vaccines, not the vaccines that we have to update every year and revaccinate people every year. What could we do that was a broadly protective vaccine so that we could vaccinate people before the pandemic starts, which is a great way to mitigate a pandemic if you can have immunity in the population before the new flu virus starts to spread. Big ambition, would be great if we could manage to get it to work. And in 2014, this Ebola outbreak happened in West Africa, and the slide just shows you the number of cases that were being counted and the exponential increase in the spring and early summer of 2014. Now, this was obviously not the first time Ebola virus had been identified. It was first found and named in 1976, probably been causing cases for a long time before that, but the first time it was definitely identified was 1976. And yet, despite the fact that there had been outbreaks, small outbreaks, somewhere in Africa almost every year since then, there was not a great deal of work in vaccine development. And the work that was done in developing vaccines against Ebola was really as part of a biodefense program, making vaccines that could be used in the event of somebody using Ebola as a bioterrorist weapon.
Just ask you to pause 1 second. Your slides are not being projected out.
Okay, we'll just wait and try and get that sorted.
Just take 1 second, I believe.
Okay, we're good now? Great, thank you. So, some vaccine development aimed at biodefense, but there were vaccines that had been tested in preclinical studies, looked extremely promising, hadn't been in clinical trials. One of these was an adenovector vaccine. It was a ChAd3 vaccine, and the only place any ChAd3 vectored vaccine had been tested in a clinical trial anywhere in the world by the middle of 2014 was at our clinical trial unit at Oxford University. It wasn't a vaccine we'd developed from the university, but we'd partnered with others to test their vaccine. And so we were contacted in August of 2014 by the Wellcome Trust initially, who wanted to help fund work in this area and get it moving forward, to see if we would be able to run a phase 1 study in Oxford, because it was felt that because the ethics committee and the regulators were familiar with the application and the trial that had been done previously, we would be able to do this very quickly and then get some initial first-in-human clinical data, reactogenicity, immunogenicity of the vaccine, pass that information on so that another group in Mali could then continue with a further trial. And to cut a long story short, that's what we did, and we did it very, very quickly. So everybody came on board and worked together, the funders, the ethics committee, the regulatory committee, the people who were doing the fill and finish, because the vaccine had not been put into vials at the time that we started this process. We had to get it vialed, tested, approved, shipped, labeled. The lawyers were the slowest here, but we finally got the trial contract signed and managed to vaccinate our first person on the 17th of September, so just actually more than a month after we were after we got involved with this, and then by the middle of November, 60 people had been vaccinated. The data was available. Group in Mali took over. Mali was next to the outbreak area in West Africa. They had had a few cases. They wanted to be ready to vaccinate their healthcare workers in particular, and it was the healthcare workers that took part in that trial. They did a very fast job as well. Middle of December, they had their data also replicating the good immune responses that we'd seen, the safety of the vaccine, and we felt it was going to go straight into an efficacy trial in West Africa because case numbers were high at that point. That didn't happen. What happened was there was a long debate about the ethical way to do efficacy trials of a disease with a very high fatality rate. And again, to keep this short, I do think that we should debate the best way to test vaccines that have a high fatality rate, but the time to do it is not when you are ready to start that efficacy trial. The time to do it is in advance of being ready to start. So we were doing our phase 1 trials. The debate about the phase 3 protocol didn't seem to start until we had our phase 1 data ready. And so what happened was it was April of 2015 by the time anybody was ready to start doing an efficacy trial with either the vaccine that we'd helped to test or Merck's viral vectored vaccine, the VSV vectored vaccine, a different technology. They'd also been going through the same process. And in the end, the way it fell out was that only the VSV vectored vaccine went into efficacy trials, and there weren't enough cases in the countries. And it was great that there weren't enough cases. They've been controlled by contact tracing and quarantine, huge public health effort to do that. But we lost the opportunity to test the vaccines because we were too slow to start. The VSV vectored vaccine proved to be very highly effective, which is great, but the bad news is that it requires ultra low temperature storage, which is not so great for vaccines that you want to use in Africa, and the manufacturing process was not mature, it was not scaled up. The manufacturing is done in roller bottles. This is not something that you can do at very wide scale, but once you have your efficacy result, you can't then go back and change the manufacturing process without relicensing the vaccine. So the vaccine that we'd helped to work on gave you the same immune response in terms of antibodies and stronger T-cell response than the VSV vectored vaccine. So we would predict that it would also be highly effective, but it wasn't tested, so we don't know. Johnson & Johnson also licensed an Ebola vaccine via the animal rule, which they did after the outbreak. But another problem is that all of these vaccines only cover Ebola Zaire, and there's also Ebola Sudan, and of course more recently we've had Sudan outbreaks, and these vaccines don't help. We knew these vaccines wouldn't help, and we knew that we would need vaccines against Sudan as well as against Zaire, but again, that wasn't done. So it was very frustrating from where I was sitting in 2014, 2015, to know that we'd done our part, we got everybody on board, worked as fast as possible, but then things stopped and we still weren't ready. And we knew we weren't ready, but we couldn't manage to get everybody together, get everybody on board to do the things that we knew we needed to do if we're going to be better protected in the future. And I think we're still in that situation now. So just thinking about the well-known outbreak pathogens, these are not the really unusual ones. These are ones that are on everybody's list. We've known about them since 1967 in some cases, since 1931 in the case of Rift Valley fever virus, and we don't have licensed vaccines for humans for these diseases, and we should have. We should have vaccines that have been tested, at least to phase 2, that are stockpiled. We have approved efficacy protocols that we would use if there was an outbreak. We also have to think about different pathways to licensure for different diseases. So for Nipah virus, for example, causes cases in Asia, very high fatality rate, actually very small number of cases. And so we're not going to be able to do phase 3 trials and test vaccine efficacy unless there's an unusually large outbreak, which we hope never happens, because fatality in some outbreaks has been 100%. It's more usually about 60%. But we have to plan different pathways to licensure using the animal rule, for example, for some of these pathogens. We know this. We could be doing this now. This is not new information. We should have plans, we should have vaccines, we should have stockpiles and trial protocols ready for all of these different outbreak pathogens that we already know about. And in my view, the best way to be prepared for the pathogens that we don't yet know about, the new ones that are going to come out of animals somewhere in the world and start causing cases in people, the best way to be prepared for that is to think about the different viral families that all of these viruses fall into. Are they coronaviruses? Are they arenaviruses? Are they henipaviruses? and make sure that we have our vaccines prepared for at least one member of each of those families. Because then, if there is an unknown virus that begins to emerge and start to cause an outbreak and maybe move to a pandemic, we have a template to follow. And what was really difficult in 2020, when we knew we had to make a vaccine against a coronavirus, is that we don't have any licensed vaccines to use in humans against coronaviruses, so we don't know what we're aiming for in terms of the immune response. There are some licensed vaccines against veterinary coronaviruses, but none for human coronaviruses, and although we knew of 4 other coronaviruses that regularly infect humans, we don't vaccinate against them because we have herd immunity against them and we don't need to vaccinate against them. So we need to be prepared for the things that we know about, and that also helps us with our preparation for the things that we don't know about. So I had been working prior to 2020 on a vaccine against a different coronavirus, MERS coronavirus, with UK funding initially, which was stimulated by our despair of what happened post-Ebola. In the UK, we got money to start working on vaccines against some of these other diseases. Zoonotic disease, camels are not the original source of the infection, but they are currently the zoonotic reservoir for the infection, and people in Saudi Arabia who go and visit camels like this without wearing a mask can get infected, and if they're not made ill themselves because they're young and healthy, they may transmit the disease to other members of their household who may then have a more severe disease. So we've been working on this virus and had generated clinical trial data, which I won't talk you through, but just to say that we'd shown we get binding antibody responses, neutralizing antibody responses, T-cell responses, everything that we would hope to see in a phase 1 trial of a vaccine against such a virus. And we also saw lasting immune responses. So this was just looking at 1 dose, but even a year after people had had 1 dose of that vaccine, their antibody responses were higher than baseline, their T-cell responses were higher than baseline. There's some, but low, cross-reactivity between MERS virus and other human coronaviruses, so it's there, but it doesn't have much impact. And all of this information was very useful at the beginning of 2020, when we found there was a new coronavirus infecting people and we needed to respond. We had also, as part of the MERS vaccine trials, planned to do a 2-dose study. This was just immunogenicity after 1 dose. We'd actually started the 2-dose study in 2020, then had to pause that and eventually stop because of the pandemic. So we'd been thinking about how to develop this further. We've been thinking about which other parts of the population we need to test in. So we had a vaccine that was proving promising. We'd been thinking about what we needed to do, and we were therefore much more ready than we might have been to put that into practice. So at the beginning of 2020, the news of the novel coronavirus came, and we sprung into action and started making a vaccine that we didn't know if we would even take into clinical trials, because we didn't know how far this disease was going to spread. But we decided as a small group originally, and then a much larger group in the university as things developed, that we would start work on a vaccine at the earliest possible opportunity, and we would keep going until it was clear that there was no point and that we should stop. And in fact, we never reached that point because the virus, as you all know very well, spread very rapidly. So we started our phase 1 trials in April of 2020. We got into phase 3 trials by June. That included 2,000 volunteers in South Africa, including an HIV positive cohort, a very important group to have in a trial. We had an HIV cohort in the UK as well. But all of this information, clinical data, from the UK, Brazil and South Africa led to the licensure of the vaccine, first of all in the UK for emergency use by December of 2020, followed by more approvals throughout the world, and then rapid rollout. So we were really fortunate to have in the university our manufacturing partners, AstraZeneca, who agreed to take on manufacturing distribution of this vaccine, and they agreed to do it without profit, because the university is a charitable institution and it requires that we are not making profits on the things that we develop. And what AstraZeneca did that was a huge amount of work, but should be of interest to you, is that they set up a distributed manufacturing network. So from one source of the cell bank, of the viral seed stock, of the protocols, of the testing strategy, this was distributed to over 25 different manufacturers in 15 different countries. And this was deliberately made as globally distributed as possible, with the aim of having local manufacturing for local supply. Now, this was set up obviously in great haste in 2020, and nobody would complain it was perfect. And in fact, what happened was that some of the manufacturers ended up making a much higher percentage of the doses than others. and then we kind of settled down on a smaller number. The Serum Institute of India had a sub-license to the vaccine. They had a very, very slightly different manufacturing process, but because of that, it was licensed separately from AstraZeneca's vaccine. Instead of Vaxzevria, it was Covishield, but that was worth doing because Serum Institute of India made half of the vaccine. So they made 1.5 billion doses, and the rest of the manufacturing network made the other 1.5 billion doses. And the Serum Institute vaccine went into the COVAX program. Again, this didn't go perfectly. That was the plan. But then in India, there was an export ban of the vaccine at some point, and we couldn't supply vaccine into the COVAX program for some time because of that. But then the manufacturing capacity was so great that the Indian need was fulfilled, and then it was possible to return to COVAX and continue with the manufacturing supply for COVAX. So another thing I wanted to point out on this map is the absence of manufacturing sites in Africa. And this was not done deliberately. It would have been wonderful to find somewhere where the manufacturer was ready to go. But as with the clinical trials, the manufacturers had to be ready to respond immediately. They had to have the facilities, they had to have the staff, they had to have the experience, they had to be able to move instantly. There was not time in 2020 to start building, to start training, to start setting up new things. So that's something that we need to respond to now, and that's great to see with the WHO-led program that is now happening. It was recognized as a weakness in this manufacturing strategy, but something that couldn't be remedied in 2020. So the university and AstraZeneca supported equitable access of the vaccine, and in fact, of the first 38 million doses administered via COVAX, 37 million doses were ChAdOx1 and COVID-19, the vaccine that we started on at the beginning of 2020. We made the vaccine available. We produced it in quantity as fast as could be produced, which was slower than we would have liked, but we got there as fast as we could. And it was low cost, and it was distributed with 4-degree storage, refrigerated storage, not frozen storage, and that really helps when you need to distribute across many countries, because that is the temperature that most vaccines are currently distributed at. So if you need to do something fast, work within the existing systems, and that's what happened. We also then began to get vaccine effectiveness data. Now, everybody focuses on the vaccine efficacy data coming out of clinical trials, but the clinical trials are in different parts of the world, in different population groups, and what we really want to know about from those trials is this vaccine effective enough to license it and roll it out? And if it is, what's the effectiveness data? And the effectiveness data that you get when you start to use the vaccine across the whole population, and in the UK it was starting with healthcare workers, but then people over the age of 80, then people over the age of 70, over 60, and so on. What's actually happening when you vaccinate that population for real? So these are not selected to be the healthy individuals. This is everybody over the age of 80 that comes forward for vaccination. And so what we were able to see then was that in the over 70s, who'd received one dose of either the AstraZeneca vaccine or the Pfizer vaccine, from 28 days after that one dose, they've got at least 60% protection against symptomatic PCR-positive disease. This is a real head-to-head comparison, so you can make that comparison there, and actually the vaccines were performing almost identically. In the over 80s, hospitalization was reduced by 80%, and deaths in the over 80s were reducing faster than younger age groups after just one dose of either of these vaccines. So this was really great to see, and the people who are working in the intensive care unit said this was a game changer. Through 2020, we had so many people in intensive care. From March of 2021 onwards, people were still going to hospital, but it was a completely different situation. It was much more like a normal flu season, and it was something that could be managed. So it made a massive difference. And then obviously vaccine rollout continued. So now I want to come to the topic of RNA vaccines. Why should we be producing RNA vaccines in low and middle income countries? I think we need better ways of responding to outbreaks, better ways of responding to pandemics. But how are we going to achieve this? Well, why would you make these vaccines in low and middle income countries? And I think we should divide that into outbreak, epidemic scenarios, or routine vaccination. So for outbreak scenarios, potentially, if you manufacture vaccines locally in country, you have rapid access, rapid deployment, ability to control the pricing. You are not in one country sitting, waiting for another country or a company in another country to make the vaccine available. They will fix the price, they will They will decide when you get it. You can do this yourselves if you can manufacture yourselves. You can have independent planning of the vaccine development process and of stockpiling needs, or you may decide that actually stockpiling isn't the best way to go. Maybe plans for rapid production when you need the vaccine is a better way to go. Maybe you should be taking the vaccine development to a point where you're ready to respond really fast if that's needed, instead of having a stockpile which then needs to be replenished, and will continue to need replenishment for as long as you keep that stockpile. So you can make those decisions yourself if you have the ability to do your own manufacturing in country. For routine vaccination programs, again, control of supply, ability to control pricing, development of a sustainable industry, building capabilities in different countries. And you can also think about the advancements in the technology that are going to be needed. Ambient temperature storage is the goal for vaccines, not refrigerated storage. Can we do ambient temperature storage? How long can we get shelf life at ambient temperatures? Failing that, can we achieve long-term stability at refrigerated temperatures? For some pathogens, mucosal delivery may well enhance the effectiveness of vaccines. If we want to vaccinate against respiratory pathogens, it doesn't make a lot of sense to me to be putting a needle into somebody's arm where we get a great response in the blood, but a much lower response in the lung. And we know from many animal studies that if we vaccinate directly into the respiratory tract, we get strong responses in the respiratory tract and really good protection. But there's a lot of work to do on how we test vaccines by mucosal delivery and how we're going to take those vaccines through to licensure. What's going to be our correlative protection? We don't yet have that for mucosal delivery. I think it's really important. It's going to take a lot of work. But there are going to be a lot of challenges. Firstly, intellectual property, which I won't say more about because we have Charles here who's able to respond to that aspect, but it's something we must recognize. There's going to be a lot of work needed. Regulatory concerns. So if you're going to want to be independent, as any country wanting to be able to make your own vaccines and license your own vaccines, you have to have the regulatory capacity to do that independently as well. And that's going to take a lot of strengthening of different countries' regulatory capacity, because at the moment many countries rely on a few regulators and then WHO pre-qualification. If you want to be independent, you have to be able to do the regulatory work as well. Distributed manufacturing can be a great way of making a lot of vaccine very quickly, but it also provides the regulators with a very complex situation that they're not familiar with dealing with and are not necessarily very happy about. And so again, that's something that has to be addressed before we need to roll out vaccines across the world again. We need to work out with the regulators how this is going to happen. And ultra-local manufacturing presents further challenges. So I have seen proposals to have a truck that will drive into some inaccessible area and then set up and manufacture the vaccine in situ and call everybody from the area in and vaccinate them, which sounds great, but you have to think about how you're going to do the quality control of those vaccines when you're doing this ultra-local manufacturing. You must make sure, for safety reasons and for vaccine efficacy reasons, that those vaccines are as well controlled as vaccines that might be made in a larger facility. And thinking back to the rinderpest eradication campaign many years ago, it was a successful campaign, but at one point, some areas that were vaccinating all of their cattle were not seeing any decline in the incidence of rinderpest. When it was investigated, it turned out that some of the vaccines from different manufacturers were actually just water, because they went through the manufacturing process, but they didn't have a quality control in place. And so if they weren't testing what they were using, they didn't know that the vaccine was actually any good. And some of the vaccines that were produced were not. So when that was addressed and the manufacturers then started to get their quality control right, that's when the campaign was successful. So we have to recognize that for routine vaccinations, if you want to make your locally manufactured routine childhood vaccinations, the prices may well be higher than the prices that countries are paying at the moment for the large Indian manufacturers producing in bulk. And that's something outside of just manufacturing concerns, that it's a political decision that countries are going to have to decide. If they want this independence, they're probably going to have to pay for higher prices. You may think that, oh, well, it'll be very cheap. We have a, local labor costs are low, but there's still going to be higher prices than the very, very large bulk manufacturing that some companies can do. So you have to take that into account. You have to think about your supply of raw materials. If you're ready with your surge capacity, you've got your seed stocks ready, and when there's an outbreak, you're going to up your manufacturing really fast. Have you got a secure supply of those raw materials? Because if this is happening in different places, if it's happening everywhere in the world, some manufacturers may have secured their raw material supply, and if you haven't, you're not going to get your raw materials, and you won't be able to put your plants into operation. So think about what's going to feed into your manufacturing process, and do you always have access to it? If we're thinking about outbreak pathogens, which is more of my area, how are you going to prioritize? Well, that's probably a local decision, but you should also take into account what other countries are prioritizing, because it wouldn't help if every country in Africa had a really great plan for Ebola vaccines and ignored everything else. So we need some coordination here. How are you going to plan for efficacy testing? Well, here I think we do need global planning, because we need global agreement on how vaccines that we can't test in efficacy trials will be licensed, because we don't want to have one country saying this is a vaccine and we know it works, so we're going to use it, and other countries saying we don't believe your evidence, we don't think you have strong enough evidence that it works. So we need plans before we start doing the clinical trials that will be acceptable everywhere, so that when those trials then happen, everybody agrees that this is an effective vaccine if it passes those tests, and it can then be licensed for more widespread use than just in the country that developed it. How are you going to plan for your rapid response? Again, I think that needs to be a combination of local and global. A lot of discussion, again, lots of input from different sources. Manufacturing facilities can't be built and then mothballed. They have to be kept warm, they have to be running, otherwise they won't be ready when you need them. And so what are you going to manufacture? If you're thinking about outbreak pathogens, what are you going to manufacture in the meantime? You could be stockpiling. When you've got all your stockpiles, are you going to make routine vaccinations? Maybe you should consider livestock vaccines, because we need a lot more livestock vaccines, and in some cases, Rift Valley fever virus, Crimean Congo hemorrhagic fever, for example, or MERS in the Middle East, if you vaccinate the animals, you can prevent transmission to humans and you can stop the outbreak ever happening. So the economic model for doing that is complex and not something I'm qualified to comment on, but I think you do need to consider, if you build these facilities, what is the best use you can make of those facilities all the time, so that they're doing good the whole time, but then they're ready to switch if you need to respond to an outbreak scenario. So I'm going to stop there. I think I've given you enough questions to worry about. I just want to end by saying that I'm now part of the Pandemic Sciences Institute at the University of Oxford, which has been formed post-pandemic. We want to draw upon the lessons that we learned in COVID-19 before everybody retires and forgets about them. And we want to be better prepared for the next pandemic than we were for the last one. And we recognize this is going to need partnership between different types of institutions and different countries across the world. We want to create science-led innovation, and we are focusing on equitable access. So if anybody wants to know more about Pandemic Sciences Institute, do look at our website. We're always happy to talk to people from different countries. And I'll stop there. Thank you.
Thank you very much indeed, Sarah. You raised lots and lots of points there. I'd just like to apologize for the certain amount of background noise that's going on, just to re-emphasize the fact that we're not just having a nice little talk on the side. There's a huge amount of actual work on development going on as we speak. So that's what the noises are about. So apologies for that. So I'd like now to introduce Dr. Andrea Carfy, who's the Chief Scientific Officer for Infectious Diseases at Moderna. Thanks, Andrea. Thanks, Sarah. That was great. Really nice overview.
So first of all, let me start by thanking Pedro LeBlanche and really the opportunity to be here and to talk about Moderna mRNA platform, but specifically the application for public health. So Moderna as mRNA platform is well positioned really to address pandemic outbreaks and pandemic response. We have been working on the technology since 2010, and already the phase trial in humans for the mRNA technology, mRNA LNP, was conducted in 2015 on pandemic H5 and H7. Since then, we've published a number of papers on the mechanism of action, but also on the LNP technology, mRNA used by us. And all that work prepared us to respond to the pandemic in 2020 with the development of the mRNA 1273 vaccine against the COVID-19. And this came together with a number of publications that really reported that the safety, efficacy of these vaccines in preclinical models, but then also in large human studies. After the pandemic, starting in 2022, we committed to develop a number of vaccines or advance a number of vaccines to early clinical studies, really against pathogens that have been identified by CEPI and WHO as the biggest public health threats. At the same time, we opened the mRNA technology or access to the mRNA technology to academic labs that are interested actually in designing and developing vaccines against those pathogens. So we provide our preclinical mRNA, the same that we use in our day-by-day experiments to labs that want to really do work in this area. And what is the concept? I mean, the mRNA is really designed to generate the protein of interest that has to be, of course, in the context of vaccines, immunogenic and raise the antibody response desired. We deliver the mRNA to the body. In the context of vaccines, we do that in the most of the cases through intramuscular injections, and then the body makes their own protein. And this is basically the cells really taking the instructions that are in the mRNA to make the protein of interest. And finally, the mRNA, which is an unstable molecule that gets degraded within 24 to 72 hours. So what are the advantages then of the technology? First of all is the biological fidelity. We are able to deliver at the same time multiple mRNA. We can express very complex molecules, and this allows also for combination vaccines, so has the potential to result in high efficacy. Also, because it's a platform, so most of the reagents are the same, the process is always the same. The only thing that changes is really the sequence of the mRNA. We can really manufacture and develop vaccines quickly. And finally, another important point is the flexible manufacturing. And we were talking about manufacturing, and mRNA allows to really use the same manufacturing site to generate vaccines against the different pathogens, but also other therapeutic interventions. So this results in a great, efficiency from a capital perspective. And this was, of course, reflected in the way we developed the COVID-19 vaccine. Since we got the sequence, within 2 days, we had designed the antigen and using our proprietary technology, the mRNA sequence. And within 42 days from sequence, we were in a phase 1 clinical study. Actually, in 42 days, we had the drug product, basically the vaccine, vialed and actually had passed all the quality control, including sterility. It took another 20 days to get into a phase 1 clinical study. Anyway, very fast timelines. And within, of course, a year, 11 months, we had approval, authorization, emergency use authorization of the vaccine in the US, and then in January in Europe. But this process, of course, which was highly collaborative and needed, of course, help also from regulatory agency and funding from governments, also not only shows the speed at which the technology can go, but also the manufacturing. Before the pandemic, we were making roughly a few thousand doses per year. By the end of 2021, we had made hundreds of millions of doses. So this speaks of the scalability and consistency of the technology. And since then, we have focused on developing a number of vaccines against infectious disease. Of course, the respiratory infections is one of the area of focus. We have, of course, the licensed COVID-19 vaccine, but now also a seasonal flu vaccine that is in a phase 3 clinical studies, a vaccine against the respiratory syncytial virus, this one for older adults, and also the combination with a vaccine that combines influenza and COVID that now is in a phase 3 clinical study. The other area that fits well the technology because of the T-cell responses is also targeting latent viruses such as cytomegalovirus, Epstein-Barr virus, herpes simplex, zoster, and HIV. So in many of these cases, these are viruses that not only causes disease in the after infection, but also have a long-term sequelae in the infected people. And of course, one area of focus is global health with a number of programs, and more recently also norovirus and the first bacterial program against Lyme. So for tackling persistent threats and emerging threats, the technologies are uniquely suited. We have, as I mentioned, committed to advanced clinical studies a number of vaccines. We have a COVID-19 vaccine approved, but we also have a number of other candidates, either in the preclinical space or already in clinical testing. So we have a MERS coronavirus that is in preclinical, but also a pandemic influenza H5 and H7 that is in phase 1. Then Zika, chikungunya, Nipah, and Mpox that are in either starting phase 2 or completed phase 2. And then a number of other additional candidates. So I will now switch to give a couple of examples of vaccines in the global health area, starting with the HIV. And of course, it's clear that there is still a need for a prophylactic HIV vaccine. With there is a list of 1.5 million incident infections every year. There is more than 20 million people living with HIV infection, and despite the use of PrEP, only 1.5 million have access to those drugs, and whereas there is a potential of 400 million people that need it. And so what is an approach to developing an HIV vaccine is really eliciting broadly neutralizing antibodies. And it has been shown that there are people infected by HIV that can develop such antibodies, and these antibodies can neutralize a large set of HIV isolates, up to 99% of global isolates. In addition, studies in non-human primates have shown the combination of these antibodies can prevent HIV acquisition if given at a sufficiently high concentration. And finally, in the AMP trial, it has been shown that in humans, broadly neutralizing antibody can prevent infection by broadly neutralizing antibody-sensitive HIV strains. So a vaccine that can elicit this kind of antibodies could actually provide protection and be an alternative to passive immunization or PrEP. So what are the two approaches that we're using? And all this work is done mostly in collaboration. So we are approaching from two different sides. One is the germline targeting that I will describe more in a second, but basically is really using an engineered immunogen to target specific B cells, and then immunofocusing, which is basically really target the immune responses against conserved epitopes, like the fusion loop in the HIV trimer. And this is a collaboration with Peter Kwong and NIH. So speaking of the germline targeting, this is an approach that actually fits well mRNA, because it requires the use of different antigens and also a number of studies in humans to really engineer the best antigen for the specific goals. Specifically, we are trying to basically use an antigen that can bind strongly the B cell lineage, the germlines that can elicit or can evolve to broadly neutralizing antibodies. And that's the first step, is the prime. And then you need a different antigen to really boost memory cells to accumulate really somatic hypermutations that are in the direction of broadly neutralizing antibodies. And as a last step is the polish, which is really giving, once that there is enough of these B cells with that can generate the broadly neutralizing antibody, use the trimer and so the polishing step to increase the titers of the response. And most of this work is done in collaboration with IAVI and is the work of Bill Schiff at Scripps that is now with us at Moderna. So we are conducting 2 clinical studies. One is with one candidate that is mRNA 1644. Here we are testing 2 different hypotheses. One is to use the priming immunogen, or the GT8 60mer, and this is really to show that we can actually engage the right germline B cells. But then we are also trying a prime boost approach where we're using a second antigen, which really is to show that we can actually increase the percentage of B cells or the B cell responses that are going towards broadly neutralizing responses. And so this study is currently ongoing. And the second study is mRNA 1574. In this case, now we are testing different designs of the glycoprotein, the envelope trimer, to see which one is the best in terms of immune responses, and the goal is to really generate autologous neutralizing responses. In this case, we are testing, for instance, a soluble versus membrane-bound, and also knockout of the CD4 binding site. And I would just pause here to say that really what we have found through our studies, that of course the design of the antigen is really critical to get the best immune responses, and some of the rules that apply to other technologies, such as a recombinant, might be different in the context of mRNA. So the second example is the work on monkeypox, and this is a pox virus. It's a double-stranded RNA virus, it's double-stranded DNA virus. There are a number of outbreaks. There was one in 2022 that was in 29 countries with 50 deaths, but there is also lots of several outbreaks here in Africa. And there are 2 main clades, clade 1 and clade 2. Clade 1 is localized mainly in Central Africa, tend to be more lethal, whereas class 2 localized mostly in West Africa and display more mild disease. In the 2022, the outbreak was more of the clade 2B, and that's really the sequence that we used for our antigen design and to generate the mRNA vaccine. So the 4 antigens that we selected were based on work that had been performed previously by Bernie Moss and the Hooper's lab of Samirid, and again it speaks of the importance of having just knowing what antigens the vaccine needs to target. And as in the context of SARS-CoV-2, we already knew that these antigens in the context of vaccinia were protective in animal models, so we could directly translate those in mRNA, but we modify, of course, the antigens to get the best responses. So for instance, for 2 of the targets, we introduce signal peptides to get them on the cell surface, remove glycosylation sites because these are made in the cytoplasm, and also remove free cysteines. In some of the others, we replace the transmembrane region, and really to increase expression while we confirmed the antigenic structure of these antigens. So importantly, these antigens are highly conserved across other pox viruses, such as variola, smallpox, and vaccinia, with a conservation that goes above 90%, suggesting that this vaccine could also work for other smallpox viruses, other pox viruses. So we tested the vaccine first in mice. This is a combination of 4 antigens. We tested 2 dose level and 1-to-1 stoichiometry compared to the human dose of the MVA, which is the modified vaccine, Vaccinia ankara, that is approved for monkeypox prophylaxis. And we compared the immunogenicity, and also we gave 2 injections 3 weeks apart, and then we challenged the animals with the vaccinia. And here are the data. So both MVA and the vaccine were 100% protective in mice from a vaccinia challenge. And also, if you look at the body weight loss, you can see that both the 2 and 8 microgram dose vaccine, mRNA vaccine, really have minor changes in body weight, and seem to be providing more protection even than MVA. Of course, mice is the first stage. Then non-human primates, when the animal model works, is always preferable. So we tested the vaccine at the 150 microgram dose compared to, again, MVA human dose and PBS control in rhesus, 2 doses of the vaccine 4 weeks apart. And then the challenge is done with actually the Zaire monkeypox, it's a clade 1, through an intravenous injection. So it's a very stringent animal model. And again, what we saw is that the animals are both MVA and the mRNA are completely protected from mortality, but also the vaccine, the mRNA vaccine, provide a really stronger decrease in number of lesions compared to MVA. Here, the red lines for mRNA, and also the duration of the disease. Basically the rash onset was delayed and was shortened compared to even MVA. So this shows that mRNA technology can be used for a monkeypox vaccine and offers an alternative to MVA. And what we are doing now is really assessing the breadth of neutralization of these vaccines against a number of different pox viruses in terms of neutralization. So this is in vitro, but also in challenge models to show that actually this vaccine potentially would be protective against a larger number of pox viruses. And we will also compare the antigens from the other pox viruses in immunogenicity study. As I mentioned, the sequence identity is pretty high, so we expect similar responses. So I will stop here. I want to just mention that a lot of the work that we do in Moderna is through collaborations, and certainly in the global health space is through an enormous number of collaboration and also support from the Gates Foundation, but I mentioned USAMRIID, NIH, and many others. And with that, I'll stop and thank you.
And next, I'm going to pass on to Robert. Robert van der Most is the Vice President for Translational Science at BioNTech. Robert.
Thank you. Thank you for having me. I changed the title and I tweaked the content a little bit because I'm going to make a slightly different pitch today, and that we haven't discussed yet. I have too many slides, and I'm still about to decide where I'm going to put the emphasis. So if I'm going to rush to a couple of slides, don't worry. Then it's all still under control. And I'll try to keep my time, but you may have to manage me. So my disclosures: I'm an employee of BioNTech. I own shares. I was a previous employee of GSK. Own shares. No other disclosures. Some safety data that I was asked to show. We'll go very quickly. Right. So let's go into the business. So we know the RNA vaccines and the basic structure we discussed. I'm not going to give you too many details, except of course the very obvious 2 points here, or 3 points really. It's flexible, it has been used, and that means we have a safety database. And I think that part applies to everything we do. It applies to us, it applies to what you're doing at Afrigen, it applies to Moderna. The safety database, I think, of this platform is an absolutely critical asset, and we can't underestimate that. I've seen my own share of safety questions and problems in my GSK time, and this is a key point. And for me, this is really the fundament that we're building upon. Now, within BioNTech, there's a few different platforms. What we all know, and what of course is the Nobel Prize-worthy finding of the uridine modification, and that changes the way that RNA signals to TLR3, MDF5, and so on. and it changes the balance between reactogenicity, tolerability, dose, and so on. So that's a game changer. And that is, I would say, probably the basis for the vast majority of RNA vaccines currently in development. I also think we need to be thinking beyond that. And the question that I want to put in front of the group here, is it possible that we have put innate immunity too far down? And one way that we're beginning to address that within BioNTech is within the TB program, and we'll discuss that a little bit later, where in fact we have the modified and the unmodified RNA. So we're bringing back the unmodified RNA on the table to see how the different way it interacts with the immune system could potentially affect its immunogenicity. Then we have self-amplifying RNA. So this has been around for a while as well. Today it's all very early, and like most of these self-amplifying RNA platforms that have been around, including in my previous company, it's based on alpha virus replicons with a subjumper promoter and a payload. For today, however, I really want to talk about the non-self-amplifying RNAs, because this is where I think 90% of the focus is going right now. So now my pitch. And this is something that, and I'm going to show you a study. It's not a BioNTech study, but it's a study with the BioNTech vaccines, Comirnaty. This is something from Bali Pulandran at Stanford. And Bali was one of the people that put systems biology on the table. And it's mean to put it very bluntly, you measure everything you can and you see what you do with it. Often said to be oversold and under-delivering. I don't quite agree with that, but we can discuss that. The point I want to make is actually coming from a discussion that I had with my mentor half a year back. It's Rafi Ahmed, based in Atlanta and Emory. And Rafael was making this point that if we take all this, I mean, everything that you see here in this slide, all the beautiful technology that we now have, literally 90% at least of current data is coming from studies being done in the US and Europe. I mean, this is the global north driving human immunology. And for Rafi, that was an incentive to try and build joint partnerships with, amongst others, India and Delhi, and they have it now because his point was there's 1.4 billion people in the subcontinent, and the level of immunogenicity that we understand is a fraction of what we know of a US population. And obviously the same applies to Africa. What the basic immunology on innate level, on all the stuff that I'm going to talk about here, is very focused, and to me that in my mind, and it's a bee in my bonnet, this is to me human immunology inequity that I think we should address if we want to be successful. And we're trying to put it in place in our TB program. And if I have time enough, I'll get into a little bit more detail there. So now about this study here. So what Bali did was a straightforward Comirnaty vaccine study in healthy subjects, immunize them, take blood samples at different time points, including the ones we know, looking at antibodies and T cells, but then also the innate ones, and looking what happens very early, in the belief that the understanding how a responder is going to differ from a non-responder, the key is in the very early innate immune response. Also understanding how in some cases we get very strong reactogenicity is there, and we need to understand that. And ultimately, for complex diseases like TB, this could be of interest to even look for correlates of protection. And this may sound like a wild idea, happy to discuss that further, but to me, there's a good incentive to begin looking at these kind of data. I'm also showing you this because we are taking this as a blueprint to, and going back to the TB study, but also a malaria study that's currently running in the US. We want to take these kind of analyses in our studies, integrate them as a way to better understand the platform itself, how it responds in the function of dose, and also how it responds in the function of the RNA modification itself. Because maybe there are cases where we need to fine-tune a little bit the innate immunity. This by itself can be a very different topic, and again, one of many questions to be discussed further. Based on my belief that everything we see today, we're at step one. I mean, this is mRNA 1.0. And in terms of further developing it, including adjuvants or other molecules that can tweak the immune response in a certain direction, I think there's way to go. And that's both the RNA and the LNP where there's room to grow. So long story short, Bali measured antibodies, and I'm not going to spend a lot of time on this slide. We know there's antibodies, CD4, CD8 T cells. What you see in the C figure, I think, was the very interesting data how a variant of concern affects the neutralizing titers induced by the ancestral vaccine formulation, and so on. So this we know, and I think this should be the bread and butter of the kind of biomarker packages we should be using in vaccine studies. This is the slide where I think we need to begin investing in generating these kind of data. I mean, what you see here is the very early innate immune response. In the B figure, you see simply changes of cell numbers, F dose 1 and dose 2. What you see in the C figure is cell types. So now all the different cell types responding to different vaccines. And then on F, what you see is serum interferon gamma going up at day 1 after vaccination. And at B, you see transcriptomics. Now, before you're asking what's the point of this, to me, there's a very interesting observation here. And that is across the board, in every figure, you see that the post-2 data are very different from post-1 data. You see the cell types in interferon gamma and transcriptomics. And interestingly, back in the GSK time, we saw something similar with adjuvanted vaccines, which is relevant because adjuvanted vaccines are still in development, for example, for TB. And there's been a lot of thinking what might be driving this. I mean, what do you change after dose 1 that then transfers to dose 2 and makes you respond in a different way? Amongst different hypotheses that these are epigenetic changes, so changes that are going to be hardwired in DNA, in chromatin accessibility, and that then change the way one responds to a vaccine. And this is hypothesis we're testing at the moment. To me, the interesting consequence of that, if that's true, if the way you respond to a vaccine depends on dose, if dose 1 changes something fundamentally, now dose 2 is very different, it affects the way that we think about prime boost, but it also affects the way, and then here I'm repeating my pitch, that immune background can be critical. So meaning, if this is what we see in Bali's study, which was done in California, if we do the same study here, we might get a different outcome. And I'm using my words carefully. I'm 100% sure we're going to get a different outcome. But I think we need to understand how that works and to what extent what you see here can be a proxy, essentially, for having different backgrounds. And then this goes back to my point that I'm quite convinced that understanding immunology on a global scale is going to be important if we want to take the tricky projects that we have in front of us and that you see in the pipeline, like TB, like HIV, like malaria, if we want them to success. So now to the pipeline that I already gave some hints about. So what we have right now is monkeypox. I'm going to be very short on monkeypox because we already seen some of the details, and the BioNTech program is actually very similar with data about to be submitted to Cell, just one antigen different. What I think is interesting in monkeypox, it also serves as an example of, or almost a model of how to respond to an outbreak. So if we take monkeypox, and right now we know it's probably not the highest public health emergency today. It might come back, and I think it's important to have stockpiling vaccines. It's also a model that if something happens, if disease X happens, and here we take it as monkeypox, how can we as a system respond, and what can we learn from what we're doing now? And to me, that builds on or it adds dimensionality to the whole COVID exercise that we've already discussed today, that was done under high pressure. Now if we do it in a different way, now we take monkeypox and we begin developing it. How do we define the antigens? How do we define the clinical development? How do we define the regulatory pathway? And to me, monkeypox is actually a way to try and better understand that. There's HSV, and I'm also going to be short on HSV. So this is a project with 3 different glycoproteins, actually done together with Drew Weissman's team in UPenn. It's in the clinic now, aimed for a prophylactic indication. So we're really looking at neutralizing antibodies, but also interested in the T-cell responses. Then we have malaria. I'm going to spend a little bit more time on that one. TB, also want to spend a bit more time on that one. There's a shingles program that's done together with Pfizer. And there's HIV, where the clinical trial, we're working hard on the clinical trial. So this is a therapeutic indication that is slightly different from what's been so far. I think it's quite promising. But I want to spend a little bit more time on TB. I mean, also because TB is of such obvious interest for this country. And what's happening is that we're trying to use the versatility and potential of RNA to develop a multiple component vaccine. Right now there's two candidates, A and B, and they differ in the RNA backbone. So one is modified RNA, the other one is unmodified RNA. And in terms of their payload, so the choice of antigens, it's exactly the same. I don't think the antigens have been publicly disclosed, so I won't do that today. Suffice to say that the, let's say, the usual suspects are all there. So where we are now is that there are two parallel studies, both with modified and unmodified RNA, both in the dose escalation design. One is running in Germany, and the other one is just starting at 4 clinical sites here in South Africa. We're getting the very early data from the German study, and that includes transcriptomics data, T-cell data, the antibody data. So very much like if you take the Bali data that just showed as a proxy, then that's actually almost like a blueprint of how we're looking at the TB study. So in Germany, we actually have the transcriptomics data like last week, which to me was very interesting because the first time that we see almost in real time how the RNA modification itself in human is affecting responses. And these responses are quite fast and very clear. And I think to see that as a dose response curve is important. And then take that same approach, because the South African study is running. There's a bit of a time gap because we started later here. But also we are generating transcriptomics data and trying to get the T-cell data, antibody data, and all built to local partnerships in the country. And once these data are available, to me the interesting part is to begin comparing, and that will be the first ever comparison of a study where we can have two very different populations with two very different vaccine formulations in a dose response setting. As you can guess from the fact that I already see like 3 or 4 variables here, the study is going to be dramatically underpowered because we can't do this in hundreds of people. But to me, as a way to begin generating hypotheses, how immune responses are developing in terms with RNA itself as a variable and different genetic and epigenetic backgrounds, to me is an important first step. So the rationale as such of TB vaccine design was to take advantage of the RNA platform, its flexibility, therefore have multiple antigens that have been targeted at different stages of the bug's life cycle. Also to have a focus on T-cell responses, CD4 T-cell response, but also the CDH, which I think is a nice and important part of the RNA platform. It's been very difficult in adjuvanted vaccines. Use the standard LNPs as they were used in the COVID vaccine and an IM formulation. And then right now begin also thinking how, and this of course is the very difficult part, and also would be part of my answer to the question on, or the emphasis on production. Before we produce anything, we have to develop the vaccine. And what we're already seeing in our discussions on TB, and none of this will surprise you, I mean, this is tough, and we're not going to be there tomorrow. And even thinking about TB efficacy studies is a challenge by itself, and we have to take the time. And here it's also that I think we need to be bullish, and we can be bullish about the potential of RNA in terms of flexibility, its production capacity, its possibility to democratize production capacity. But in the end, when we're down to development, I think it's still old-fashioned hardcore development. And we need time, and we need numbers and study designs that will give us the kind of de-risking or even efficacy data that at the end of the day we need. And that's where we are with TB. Malaria is slightly different. It's complicated, but I like to think it's an order of magnitude less complicated than TB, amongst others because there's a human challenge model. And there's a well-recognized antigen, the circumsporozoite vaccine antigen, which is in the vaccine. The way that was done was to, and that I think is, good illustration of the flexibility of the RNA platform. I think the preclinical folks made about 40 constructs, tested everything in mice, did challenge studies, acknowledging the mouse model is not perfect for malaria, but it does give an indication and maybe an ability to rank things. Measured a lot, and we did finally put the data in a principal component analysis to see what in the end was promising, choose a candidate, kept on developing, choose another candidate, And right now, one candidate is in the clinic, the other one should be in the clinic any moment. We will look at we had a look at the very first data, and again, I think a key word here, it's collaboration. I think this entire field hinges on collaboration, so we're working a lot with Georgia Tomara's lab on getting the kind of antibody functionality data here in terms of avidity, functionality, and so on. And then make an informed choice as to what we put in a human challenge model, and this will be the classical type models being used extensively in RTSS, whilst at the same time beginning to think how and when we can bring this to Africa. Should we wait for the human challenge model or shouldn't we wait? Should we go for a seasonal area or perennial area? How big should the study be? And again, this is very much ongoing discussions at the moment, but there's a clear intent and commitment to take this vaccine into Africa, unless something really a catastrophe event, what I call it, happens along the way that really immunogenicity is bad, or efficacy in the human challenge model would be very disappointing. We're trying to plan, of course, for success here and see how we can move forward and plan accordingly. So what we're also planning beyond the serum sporocyte protein is to see what other malaria or Plasmodium antigens are there. And of course, there are many of them. And what we've done there is to try and make what we call a T-cell string to identify chunks of proteins of interest, identify the hotspots for T-cell antigens, stick them together and put that into a separate vaccine design that would then go as a second RNA molecule together with the optimal one for the CS protein. So this is moving forward now. Then we have HSV, which I already briefly touched upon. And here you see the three proteins that we're currently looking at, so GD, GC, and GE, all for HSV2, and right now all for a prophylactic indication, although it hasn't escaped our attention that a therapeutic approach to HSV is of quite some interest as well, but we don't know, and again, this is topic of debate, whether it should be the same formulation or whether we should identify different antigens, different T-cell antigens, and do this in a separate or distinct approach. There's monkeypox, both as a way to respond to the outbreak that happened, and also to see to what extent this can be a model for pandemic response pathways. This is being done together with CEPI, and for me, I think I've spent some time at CEPI, and I think that's a very positive collaboration. The antigens are shown here. They are, I think, 3 out of the 4 overlapping with what Andrea presented. We have one unique. What's interesting to know about this vaccine is that monkeypox has two variant shapes: the mature and the EV, MV and EV. They both have different antigens. So EV has another envelope with specific envelope proteins. And we selected to take 2 EV antigens, 2 MV antigens. That's currently being evaluated in mice. It's very protective. In non-human primates, it's also very protective. That whole package is being submitted for publication and is about to go in the clinic or might already be there at the moment. Which brings us to the pipeline as is. So there's a respiratory virus pipeline. And this is very much in collaboration with Pfizer. So the original Pfizer partnership is still very much there. And either combinations with other pathogens like RSV, like flu, are all in that bucket. And there's the shingles program. There's the HSV program that is a BioNTech program only. So shingles being VZV and HSV is really the herpes virus area. And then what we call the global health area, and that today is malaria, TB, and monkeypox, and to some extent HIV, although the HIV program will start in the US, but also with the intent and commitment to bring it here. So in order to further build the required partnerships, something called the Global Health Office was started, led by Pedro Alonso, who will also be at the meeting 2 days from now. And then really the idea is that simultaneously build the required networks and decide how we're going to move forward, and do that in parallel with the clinical development as is. So designing clinical studies, designing biomarker plans, just get it done. And then also keep thinking on the on how we produce what and where. So summary. So what's clear is that the technology is a game changer. We can be fast, it's shown to be protective, and we have the safety database. What we see now from preclinical data and very early clinical data is that it can in fact be applied to other pathogens. And I think this is opening a huge box. I think it's a very interesting, I mean, for BioNTech, this came from oncology. So to me, the developments in oncology and how that can be seen at a global level is an interesting discussion, but also beyond that. And one of my personal favorites is autoimmune disease. Is it possible to tweak the RNA LNP platform in a way that it can be tolerogenic? I think it's very early days. There's some very early data from BioNTech in a mouse model that with lipoplex, which is not an LNP, but it's a different form of lipid-based structure, it can be done. I mean, admittedly, very early data, but to me, it could open an interesting door. So what we're doing now, our focus beyond the oncology area that I did not touch at all, we're developing, we have these vaccine candidates in the pipeline for different critical infectious diseases with high unmet medical needs. So I say that the big three are there, which reminds me of the early days of GSK, where GSK then found out that actually this is quite difficult. So I suppose we're going to find the same thing. But what I do like to think is that areas of systems immunology and also what's the buzzword of precision immunology can help us and maybe come to earlier de-risk or fine-tuning of the system. And that way, by coupling immunology with the flexibility of RNA design, maybe we can come to earlier de-risks or different decisions and earlier changes. And of course, those kind of studies and that kind of intent, in my view, is a very good starting point and breeding ground to build the collaborations partnerships we need in development, hopefully also in production. And this is a very different topic and my final slide. As you probably know, there's the concept of BioNTainer, which is a local mobile modular production unit. It's based on 2 sets of 6 containers. So one makes the drug substance, the RNA, the other one makes the drug product with the LNP. Fill and finish is not included. It's early days, I would say. It works. It works in a GMP type way. Right now, the planning where BioNTech is to have one of them in, as you undoubtedly know, in Rwanda, in Kigali. So that's there, and it should at some point be opened. What Sarah mentioned, I mean, how you keep them warm, I think that is a critical issue. Now the choice is to make Comirnaty because that's the only thing that can be made because it's a licensed vaccine. Beyond that, obviously, everything is in research quality, which is not a bad thing because I think with the research and R&D and development potentials we have, the capacity to make clinical grade RNA molecules is going to be critical. So to some extent, the interest will be in really commercial production, but in my mind also in early clinical R&D production. And with that, I give the floor back. And thank you for your attention.
Thank you very much, Robert. We are running a little bit behind time. We've got one more talk, though, from Amin Khan, who's the Chief Science Advisor to Afrigen. And I think Amin is online, I hope.
Online. Thank you, Charles. Can you hear me? Charles, can you hear me?
Yes, thanks.
Thank you.
We can see your slides too.
Thank you very much. For some reason, I'm locked out of operating my video, so you won't be able to see me. So first, it's a pleasure to be here today to represent Afrigen at this colloquium. Many of the aspects have been discussed already by the previous speakers with respect to the opportunities that mRNA technology presents, especially for low and middle income countries. I won't dwell on those very much. Today, what I would like to do is to present the approach that Afrigen is utilizing in establishing the technology, not only from the point of view of preparedness, but we believe very importantly for sustainability, to have a network that is focused also on being able to do research and development. So next slide, please. Afrigen was created to establish production capacity for the African continent. And those of you who are in Cape Town today recognize that this is obviously in the southernmost point of the African continent. It's been a pleasure to be associated with Afrigen over the past year as they continue this journey. The adjuvant formulation, which won't be discussed today, was established already at Afrigen at lab scale and is being utilized. The end-to-end mRNA vaccine technology development and manufacturing, which is the subject of today's discussions, has established the fundamental capabilities that those of you who are present in Cape Town will, I'm sure, have the opportunity to see. And these technologies have been demonstrated successfully in preclinical work, and the organization is now preparing for GMP certification in the coming few months. Next slide, please. To establish the fundamental technology here, Afrigen have utilized AfriVac 2121, which is a SARS-CoV-2 vaccine candidate, to put all the pieces in place to enable the manufacturing process development, the analytical sciences and release testing capabilities, the methods and capabilities, and also the LNP production. And ultimately, the intent here with this using AfriVac 2121 is to demonstrate the successful implementation of this platform, which, as you know, is being tech transferred to the partner organizations across the globe. Next slide, please. Oh dear, I think there was a slide missing. Nevertheless, so as I've mentioned, to establish this, the organization is focused on the systems and processes that are necessary to enable GMP pharmaceutical vaccine manufacturing, testing, and release. And they're shown on this slide at a very high level, from initially setting up a quality management system, putting in place the laboratory capabilities to do plasmid design and development and process development, and ultimately, obviously, putting in place the GMP manufacturing process and the facilities. And actually, Afrigen have a couple of technologies here that can be applied to mRNA LNP. Ultimately, one wants to be able to release GMP material, and having appropriate quality control systems in place is obviously critical. Now, unusually for a small entity such as Afrigen, I want to point out that there is also work underway to establish aseptic fill finish capabilities. So Afrigen will have not only the basic capabilities to manufacture mRNA, to produce LNPs, but also produce the final filled vials with the vaccine product. Next slide, please. So here, at a very high level, I've already discussed this, but let me just reinforce the point. AfriVac 2121 is utilized to establish the manufacturing processes and analytics. That has been done. And the physicochemical quality has been tested of AfriVac 2121 versus a commercial comparator vaccine, demonstrating that both the physicochemical quality and subsequently the preclinical immunology and efficacy in suitable animal models is comparable to an established COVID vaccine mRNA product. And the company is now well underway to establishing the capabilities to do GMP manufacturing and release testing. And subsequently, there is opportunity to conduct phase 1 clinical studies to demonstrate the ultimate performance of the platform from a human safety and immunogenicity standpoint. Next slide, please. Now, it's been pointed out by the previous speakers how important it is to have capabilities in the various areas that are required for successful research and development for mRNA. And the approach at Afrigen at a very high level is relatively straightforward. Already mentioned, one needs to establish manufacturing technology. But as we all know now, having a GMP manufacturing facility that is standing ready for pandemic preparedness is not a solution for sustainability. What does that capability, that capacity do between pandemics or the emergence of pathogens? To enable that, one has to focus on building the broader capabilities, the infrastructure required to do the research and development work for local or regional targets during the interpandemic periods for routine vaccination products, as mentioned earlier. So the manufacturing technology is being established. There's formulation research work that is ongoing, not only for mRNA, but more broadly for vaccines. The activities to support the nucleic acid, not only manufacturing, but design is critical for long-term success. And as I mentioned earlier, important to apply these approaches and these technologies for vaccine targets that are going to be critical for local or regional routine applications. And as mentioned earlier, one needs to continue to focus on optimizing formulations for cold chain, so making them more stable, or even alternative delivery methods going beyond intramuscular injection. So, as I said, it's important to not only prepare for response to pandemics or regional outbreaks, but also to utilize these capabilities and facilities to focus on disease targets that are of local and regional interest. Next slide, please. So earlier this year, actually during a meeting in Capetown, where the various, all of the partners came together and discussed what the needs are for their regions, here is essentially a summary of what was proposed, and this is a slide taken from Pierre Gazelle. And as you can see here, it's a broad cross-section of disease targets, not only for viral diseases, but also some bacterial diseases and some other pathogens. The ones that I've highlighted in red are the areas where Afrigen has begun to play a very significant role in applying mRNA technology, working with partners for these disease targets. And I won't spend too much time going through the details of these, but next slide, please. Highlight just two of them. And the point that I want to make here is that it requires a collaboration with a broad set of partners, bringing to bear very specific capabilities and capacity to enable the continued technology development in these critical areas. Here I'm showing the Rift Valley fever virus vaccine. This is a project that was initiated recently. It's a very broad collaboration, and the collaborators are shown on the right panel at the top there, not only for the clinical aspects that one will need to, capabilities that one will need to have in place to do the early clinical testing, but also the regulatory, the manufacturing, the aspects of RNA design and formulation, especially the desire to make more stable formulations for LNPs. So this is an example from Rift Valley fever. As I said, this is a project that was initiated recently. Next slide, please. And another example here is RSV vaccine. And again, on the top right-hand panel, you will see the various partners who are a part of this collaboration, this consortium of partners who bring to bear their specific capabilities to enable the development of a RSV vaccine for LMICs. Next slide, please. In fact, Afrigen have looked very broadly at all of these areas for potential partners and collaborators, from RNA development, DNA plasmid design, manufacturing technologies, various delivery methods, continuing to focus on formulations research, and the very specific targets of regional and local interest. And here what this slide is depicting is each check mark shows a collaboration that is either already in place or is imminent to continue to drive the capability, capacity development in partnership with Afrigen to focus on LMICs. Next slide, please. Everyone now recognizes that for sustainability and to continue to build local capability and capacity, there needs to be an open-minded approach to establishing this mRNA R&D ecosystem with a focus on LMICs. And I've highlighted there in red the antigen design piece, which is extremely important. And here I'm not talking about antigen discovery, I'm talking about optimizing the antigens for applications to various disease targets here from the point of view of applying mRNA technology. And it's an area that Andrea Coffey is adept at, and Afrigen are beginning to establish collaborations that will facilitate the continued work here as part of the network. Next slide, please. In summary, and I won't go through all of these bullets, I think these were discussed before by the previous speakers, the potential for mRNA technology, the lower barriers to entry for LMICs applying this technology. But there is a need to continue to develop the partnerships and to bring to bear efforts to continue to establish and elaborate local capabilities to build these capabilities and capacities to apply for local and regional application. As you see, the strategy is relatively straightforward, and it is to build a platform using SARS-CoV-2 vaccine as a prototype to demonstrate in the clinic that it is indeed safe and produce an appropriate immunological response, improve the platform through selective collaborations and broad collaborations, and assure freedom to operate, a topic that I didn't dwell on today, but it's critical for continued success here. And to, importantly, apply the platform through collaborations for local and regional vaccine targets. And next slide, please. None of this has been possible without the continued support through partners that are shown here, collaborations, funding sources, and in particular, the support of the WHO and the MPP. With that, I conclude my presentation, and thank you very much for the opportunity.
Thanks very much indeed, Amin. Okay, we are what would normally be closing time here. However, I would like to take a few questions. Does anybody need to run at this particular moment, or have you got 15 more minutes that you can stay with us? Good. Yeah. So there have been a lot of questions that have come in online. So I'm actually going to start with Martin Frieder because we haven't heard from him. Martin, are you there?
I'm here, Charles.
You are, great. So Martin, thanks very much for joining us. What do you think are the kind of science and tech innovations that we need to make sure that this platform really has the public health impact that we need.
Okay, so thanks, Charles, and to everybody on participants and online. Apologies, I can't be there. I think if mRNA could solve the problems of delayed flights, that would be the biggest public health innovation we can do. So I leave that to you. Okay, so the innovations that we really need to see, I see we've got multiple parallel tracks here. So the objective of WHO and MPP setting up the mRNA hub was first and foremost about pandemic preparedness. This is something that Sarah addressed very well. And so this is a tool that we could use in the future. The hard part is going to be two things. One, getting the price and programmatic suitability down. Currently, it is not programmatically suitable in the LMICs, and certainly in the hands of Afrigen and the rest of us, the cost per dose is kind of bordering, it's above $5. This is too expensive. So we need to improve the technology to make it thermostable, bring the price down and make it immunogenic. But we also need to understand better how the nucleotides influence this. Robert touched on this a little bit. We know that Drew Weissman and others are working on this. This is critical. How do we best induce antibody responses? How do we best induce T-cell responses? So understanding the tool better is critical. And then at the same time, for this to be sustainable, we need to identify the application to products that can be sold in the LMICs. Now, again, if we don't bring the price down, this is going to be difficult. I also want to remind people that just because you can make a vaccine, it doesn't mean that the government is going to procure it. The budget available for many public health interventions is getting increasingly tight. So we need to be looking at what the burden is. And this is, I think, where therapeutics are going to come in and veterinary vaccines. Now, most of our mRNA collaborators don't have a therapeutics approach. So this is where in-licensing and collaborations with groups like Moderna, like BioNTech, will become absolutely critical. I just like to also come back to something Sarah spoke a lot about, what we can do with mRNA. I also do want to highlight that mRNA is a tool. We need to not lose sight of the other tools, such as viral vectors. And we have just briefly touched on mucosal immunity. We know that for some applications, mucosal immunity is likely to have a critical role in not only preventing infection, but preventing further spread of the virus. So it could very well be that mRNA will do the job. We don't know, but let's not lose sight of the role of viral vectors in this. So Charles, I'll stop there and pass it back to you.
Thank you, Martin. Sorry, are you Sarah or Sarah?
Sarah.
Well, I got a question for you, Sarah, from civil society. And is civil society part of the Institute, given our work on innovation and access, given their work on?
Thank you. So the Pandemic Sciences Institute in Oxford was born out of the success that Oxford University had in responding to the pandemic, which came from different areas. So we developed the vaccine with AstraZeneca. Peter Horby ran the recovery trial, which tested existing drugs in hospitalized COVID patients to find out as quickly as possible with very simple but very large studies which drugs work and which drugs don't work. And they reported on both of those outcomes very, very rapidly. We also had many other aspects of pandemic response work going on, serology testing, some early work on diagnostics, which didn't really come to fruition, developing an app, and various other things. But what we recognized as we came to form the Institute is that we were missing a social science input into this. We were missing the kind of people's responses to pandemic interventions, be they vaccines, be they non-pharmaceutical interventions such as lockdowns. And so we invited social scientists from the humanities division to come and join us. And to think about how we can respond better, how we can engage with different parts of society, also in different countries, to have a broader response, not only thinking about the interventions, but also how people receive these interventions, because we can have the most effective vaccine in the world, but if nobody wants to be vaccinated, it has no impact. So that's a work in progress. We have put a lot of emphasis into public engagement, into providing clear and accurate information in as many formats as possible, particularly about the vaccine, throughout the pandemic period. We recognise, though, that needs to be a two-way process, and this is something that we're building on and hope to do more of that in the future.
Great, thank you. I've got a question for you, Andrea. Someone called Shamsuddin, who's a student, has said that he's curious regarding difficulties with the filtration process involved in mRNA production, especially when you get up to large scale. And what are the current strategies on the ground to improve the filtration pipeline so as to enable large-scale production?
Yeah, I'm afraid I can't really answer to that. Not because I don't want to disclose it, but I'm more in the research, not in the development side of the. I mean, this certainly I can say that filtration has been an area of focus. By coming to large scale and how we have addressed it, I don't have much to comment. I just don't know, honestly.
I'll take a stab at that since we are in the throes of solving our filtration problems. I think really, really important, what we've seen is as you go from small scale to large scale, the varied differences that you see, and we've certainly had some issues as you transition from one particular buffer to the next, and also it's dependent on the concentration of mRNA. So we haven't solved the problem. I can just say that we solved our initial issues, but really important for the likes of like Afrigen to also participate in this translational space as you go from early work to late stage scale up development. And everybody thinks about the large scale manufacturing, but how do you solve these little scale up problems? And so we've really been able to see that you have to be able to do the development work through the different scales. So yeah, I'll stop there.
Okay, thank you. This is another question for you, Karen, but you'll probably just throw it back at me.
Maybe.
It's, what is the IP management plan for the hub, considering the potential IP infringements, and how is this going to be implemented in different developing nations where the IP position may vary?
Yeah. So I'll take that one first, and then Charles, you're very welcome to add to that. So, I mean, as we all know, the IP space is very, very complicated, but one of the sort of objectives within the program is that we are moving towards collaborations and technologies that take us outside of that space so that we have freedom to operate. And there's a very complicated model around multilateral technology transfer, which the MPP is managing, and also looking at where patents are being filed across the different areas, particularly in low middle income countries. So there's not one straightforward answer to this one. And I think that, in a global access program, it is complicated, but the idea is that for the hub and for the partners that are in the hub to ensure that we have the freedom to manufacture these vaccines and get them to people that need them. Charles?
I'll just add to say this is one of the reasons that WHO asked us to partner on this program was really our expertise in licensing and patents. And so we have this ongoing mapping that we're doing, both more broadly with new patents coming out that could impact, and then more focused with each of the individual partner organizations, supporting them to do detailed freedom to operate analyses in their countries. But clearly the idea is that we will not be infringing patents, and we can invent around them, we can license. Actual company, to be honest, there are a number of strategies that you can, there was talk at the beginning of this, really a malicious rumor spread that we were going to be infringing patents left, right, and center. And this is absolutely the opposite of what we're trying to do. We've probably got time for maybe one or even maybe two questions. Okay, this is for anyone can take this. For LMICs, the major challenge is the cost of tech transfer of the mRNA technology, as there is no or little R&D support and funding. Anyone want to have a think about the R&D ecosystem?
Well, I'll have a first stab at it. I mean, I think the idea between the 15 hubs that WHO is setting up in the network is so that this will minimize the number of times technology has to be done independently, and therefore look for economies of scale, if you like, sharing new developments as quickly as possible, and therefore reducing the cost. So if we all collaborate, instead of trying to work on siloed collaborations with just one LMIC manufacturer and one maybe a company that owns IP, that's not the way to do it. What we should be doing is looking to work more broadly with IP that come and R&D know-how that comes into the hubs then being shared, because then we can reduce the costs.
Great, thanks. Okay, and now it's time to put you two gentlemen on the spot. So you very kindly come to this event. Thank you. How do you see potential ways of working with the program from your perspective? That clearly you have, your own commercial strategies and so on, but how do you see this, and what are maybe tentative next steps to see what could be done together? I don't know which one of you wants to start.
I'll start with maybe a couple of things. One is and it was alluded at the beginning, there is one aspect is really finding way to get these vaccines, especially for emerging infectious disease or outbreaks, a way to get them licensed, right? That remains, a challenge. Of course, there is many different ways to do that. Discussion generally starts after there are at least, clinical data with the regulatory agencies. So that remains, a hurdle for companies to see how to bring those molecules forward. And then I think for me, it's a partnership model, right? There has to be, these are clearly, in many cases, are not vaccines that are, bringing profit to the companies and of course to the investors. And so there has to be a model where there is a partnership between the industry and the public sector to really making them available to people, conduct studies and so on.
So I'm going to give the horrible answer. It depends. And it depends on what we truly want, because I'm deeply, I really think that our problem right now is a development bottleneck. It's for many things not a manufacturing bottleneck. And that means if we would rush too fast in establishing manufacturer spots, in any kind of way, many hubs or many biocontainers or combinations thereof. And this is a question I've been asking in biotech for a year. What is it going to make? We can make Comirnaty, that's clear. And in Kigali, Comirnaty will be made. This will keep it warm, and maybe there's demand, and we're good. That's fine. But everything else is going to take time. And to me, the question on how we can jointly get there, I like, and maybe it's a naive thought, it's part of it is simply by doing it. And for me, the fact that we have these ambitious programs to have a clinical development plan here in Africa, and if that is full speed, and it is TB, HIV, and malaria, that means that there will be a massive need to build partnerships on every aspect, on clinical sites, on regulatory discussions, on biomarkers, how to do immunology. Particularly in the latter one, it's at least a vision of my own team that we build the partnerships here. So we do immunology in Africa, and we do it in depth, the way we can do it, and so on. And that simultaneously, while doing that, there's going to be a discussion on how to make it. But right now, discussing who's going to make the TB vaccine for me is a theoretical point, because we don't even know if that vaccine will work. Maybe M72 is a success, and we have a very different discussion. And to me, that's also a point that we haven't discussed at all at the moment. And to me, would be an answer to what Martin was saying on cost, because there's cost of goods and there's cost effectiveness. And I was very happy to see on Amin's slide the Q1, the company on that can do the health economics and modeling, because there's some beautiful data on the health economics of a TB vaccine, which depends on efficacy. And to me, then we're back to development. And that's where I think we should focus and build the partnerships. And where I'm optimistic, and that would be the short answer, is that as we go along, and as we do this in the partnerships that we're building, I would like to think that that is enough to build the trust and get to know each other, to then begin thinking how we're going to do that. I mean, let's assume that the TB vaccine works, or that the TB vaccines being developed at the RNA hub works. How we're going to do this? How we're going to make 500 million doses? I mean, that's a different discussion. I think we're very far from that, but I think to begin building the kind of partnership and dialogue to prepare us for, if that day comes, how we do this, I think that to me would be the way to go.
Okay, thank you. The lights were telling us it's 4 o'clock. Basically, they come on, say time to leave. Karin, would you like to say a few final words before I hand over to Martin?
Well, there's a lot of things to say. I would like to say thank you very much to have been invited on this esteemed panel, and it's such a privilege to be sitting with you. I think the discussions today and the talks that we've heard have shown that we've just really started working on mRNA development, and there's a lot to be done. And I think that if you remember Amin's slide, which was the rainbow, that he showed that there are going to be great advances in the cross-cutting technologies. For instance, when we look at mRNA and adjuvants, or novel formulations and alternate delivery methods, and this is where the real innovation is going to take us into addressing unmet health needs. Thank you.
Thank you. So please join with me thanking our fantastic panel, and also those who contributed online, for a very interesting afternoon. Thank you very much indeed. And thank you very much indeed, Petra, for hosting us, as ever. Martin, did you want to say a few words finally?
Okay, really, I think, Charles, just as you, to thank everybody, to thank Afrigen for hosting this, to thank Afrigen also for hosting the hub and trying to help the low and middle income countries. Thanking BioNTech and Moderna for the excellent innovation and what you've been doing to bring these vaccines forward. I think we are at the start of a big quantum jump in medicine, and it's great to be there. But let's not forget that recombinant proteins, it took a long time between the first product and the second product. So really, I hope that we can partner with you in bringing these forwards. And Sarah, I really want to emphasize that there's a role, and I hope we can actually bring a parallel activity on viral vectors, trying to do for viral vectors what we've been trying to do here for RNA. So to all of you that are in the room, that are online, thank you very much, and bye-bye.